Hello students, let us do the quick revision of 40 important diagrams from chapter 2 to 6 as diagram based questions are likely to come in your first term exam. This is the first diagram. It is diagrammatic view of a typical Anatropus ovule and you have to label A, B, C and D. A is the funicle, a stalk with which ovule is attached to placenta. B is micropyle. It is a small opening through which pollen tube enters inside the ovule. C is the nucellus inside which embryo sac is present, and D is the chalazial pole of ovule which is opposite to micropylar pole. Second diagram, it is a typical dicot embryo, means embryo with two cotyledons. In this, A is plumule or stem tip, B are two cotyledons which store food, and C is the radicle which develops into root. Third diagram, this is Gynosium representing female reproductive part of a flower of papaver, where more than one pistil are present and the pistils are fused together. So, it is multicarpellary and syncarpous pistil. Here in the diagram, A is stigma and B syncarpous ovary. Fourth diagram, it is the structure of pollen grain which is haploid and it is at two cell stage. Now see the parts A, B, C and D of pollen grain. A is exine. Pollen grain is covered with hard outer layer exine made up of sporopollenin, and B is intine, which is inner layer of pollen grains made up of cellulose and pectin. C is a vegetative cell and D is a generative cell which later will form two male gametes. Diagram number 5. It is the enlarged view of microsporangium present in the anther. One anther has 4 microsporangium. Now label parts A, B, C and D. Microsporangium is generally surrounded by 4 layers. A is epidermis, B endothecium, C middle layers and D tapetum. This tapetum nourishes the developing pollen grains. Also, cells of tapetum may have more than one nucleus. So please remember these 2 points about tapetum also. See the diagram number 6. Here, first diagram is of fertilized embryo sac where A is zygote which is diploid and B is primary endosperm nucleus which is triploid or 3N. Now see the second diagram. It is showing stages of embryo development from zygote. Zygote develops into C, the globular embryo stage. D is the suspensor present in heart-shaped and mature embryo, and E are cotyledons. This is the 7th diagram. The 3 diagrams here show the part of ovule. See all the 3 figures and check the questions A to D. Question A. Cell division in diagram marked A is 1 megaspore mother cell towards micropylar end of ovule undergoes meiosis division. So it is meiotic division. Question B. 2 cells formed in B. As a result of meiosis 1, 2 cells are formed and this stage is called megaspore dyad. Question C. 4 cells marked C in the figure. This is megaspore tetrad formed after completion of meiosis 2. Now what happens to cells marked C? Here, out of 4 megaspores, 3 degenerate and 1 is the functional megaspore which develops into female gametophyte. Now come to D question. Total process is called as, megasporogenesis. 8th diagram, you have to identify types of pollination in given plants. A is the wind pollinated plant with compact inflorescence and well exposed stamen. B, it is water pollinated plant Vallisneria where female flower reaches the surface of water by long stalk and male flowers release pollen grains on the surface of water. C is an insect pollinating plant where flowers are large, colored, fragrant and rich in nectar. Ninth diagram. It is the longitudinal section of an embryo of grass which is a monocotyledonous plant, and you have to label A, B, C and D. A is scutellum, which is the cotyledon. B is coleoptile, which covers shoot apex. C is epiblast, which is a flap-like structure present opposite to scutellum and which is continued with the coleorhiza. D is coleorhiza, which covers root apex. Come to 10th diagram. It is also a monocotyledonous seed of maize. Now, note the parts A to F. A is pericarp. B, aleurone layer, which is the outermost layer of endosperm. C is endosperm, which is starchy. D is scutellum. E, coleoptile and F, coleorhiza. This is 11th diagram. Here, karyogram of a person is given and you have to tell the disorder the person is suffering from. This person is suffering from Klinefelter syndrome with XXY condition. Here, instead of 46 chromosomes, 47 chromosomes are present with one extra X chromosome. Now, see the karyogram of the person in 12th picture. Here, the person is suffering from Down syndrome again with 47 chromosomes. Trisomy of 21st chromosome can be seen here, and that led to the disorder called Down syndrome. 13th picture is a karyogram of a person and you have to tell the sex of the person. Here, the sex of the person is female, which can be identified due to the presence of two X chromosomes. Now come to 14th picture. It is showing the disease sickle cell anemia, where the shape of RBC changed to sickle shape instead of biconcave disc shape. It is an autosomal linked recessive trait. 15th picture. Here also check the karyogram. This person is suffering from Turner syndrome with condition XO. Instead of 46 chromosomes, 45 chromosomes are present in this person. Note, there is only one X chromosome instead of 2. Come to 16th picture. It is showing symbols used for preparing pedigree chart. Here, A, B, C and D are. A is used to represent affected individuals. B is used for mating and C is used for identical twins, while D is used for fraternal twins. 17th picture. Here is a pedigree chart of a family for a particular trait. Identify A. Is it autosomal or sex-linked trait? It is an autosomal trait as it is expressed both in male and female. B. Is it dominant or recessive trait? It is a recessive trait as normal parents of second generation are having affected child in third generation, or you can say there is a skip in generation in this trait, so it is a recessive trait. 18th picture. Given here is a cross of homozygous plant. You have to tell genotypic and phenotypic ratio of F2 generation. As it is a monohybrid cross, the genotypic ratio of F2 generation will be 1:2:1. 1 capital T, 2 capital T small t and 1 small t. And phenotypic ratio will be 3:1, where 3 are tall and 1 is dwarf. 19th picture. This picture is showing genetic basis of blood groups in humans. There are 4 types of blood groups: A, B, AB and O. Now answer the question number A. Number of alleles involved in blood grouping are 3 alleles are involved in blood grouping. These are IA, IB and small I. Thus, it is an example of multiple allelism. Answer of question B. A person having AB blood groups has both alleles. What is this inheritance type called? It is called as codominance, as both alleles IA and IB express their own types of sugar. 20th diagram. The given picture is showing the experiment performed by Frederick Griffith in 1928. He worked on bacteria Streptococcus pneumoniae and witnessed a miraculous transformation in the bacteria. Now see the results of the experiment. In picture A, when rough strain R was injected into the mice remained alive, as R strain is non-virulent type. In B, when smooth strain was injected into the mice die from pneumonia, as S strain is virulent type. In C, when heat killed smooth strain was injected into the mice was alive again. And in D, when mice was infected with mixture of rough strain and heat killed smooth strain, the mice died and he recovered living smooth bacteria from the dead mice. So, he noticed transformation of bacteria in this experiment. 21st picture. See the steps of experiment and answer the questions A to D. A. The objective of the experiment was: this experiment shows semi-conservative replication of DNA. B. This experiment was performed by Michelson and Stahl in 1958. C. He performed this experiment on E. coli bacteria. And D. Very similar experiment was performed by Tyler on Vicia faba in 1958. 22nd picture. This picture is showing another experiment performed by Alfred Hershey and Martha Chase in 1952. They proved that DNA is the genetic material through this experiment. They worked with viruses, bacteriophages that infect bacteria. They used radioactive phages. In one case, S35 was used to make protein coat radioactive, and in other, P32 was used to make DNA radioactive, and it was found that DNA is the genetic material and not the protein. 23rd. Here is the picture of Lac Operon. In the picture, what is A doing? A is lactose, which binds with repressor protein and inactivates it. Thus, repressor is now not able to bind operator and Lac Operon is on. Next, label B, C and D. B is beta-galactosidase enzyme, C permease enzyme and D trans-acetylase enzyme, and genes responsible for their formation are Z, Y and A respectively. 24th diagram. This is the structure of transfer RNA. The secondary structure of transfer RNA looks like a clover leaf, where A is the site for amino acid binding and B is the site where anticodon is present, and also the compact molecule of transfer RNA looks like inverted L. Now come to 25th picture. It is a schematic structure of a transcription unit, where A is the template strand, B is coding strand, C is promoter, and D is terminator. 26th. In this picture, you have to identify 3 events A, B, and C. A is transcription, where primary transcript RNA is formed from DNA. B is processing of primary transcript to mature transcript, which involves splicing, capping, and tailing. C is translation, where protein is formed from messenger RNA. 27th. This diagram is of a human ovum, where A represents cells of corona radiata and B zona pellucida. 28th. This is the table showing codons for the various amino acids. A. Mention the nonsense codons. The nonsense codons or stop codons are UAA, UAG, and UGA. B. Which codon initiates translation? Does it has any other function? The initiation codon is AUG. It also codes for methionine, so it has dual function. C. Genetic code is degenerate, as some amino acids are coded by more than one codon. For example, serine SER is coded by UCU, UCC, UCA, and UCG. 29th picture. This structure is of blastocyst, which gets implanted into the uterus. In the diagram, A is trophoblast, which gets attached to the endometrium. B is inner cell mass, which gets differentiated as the embryo, and C is the blastocele. 30th picture. It is the structure of a nucleosome, where A is H1 histone and B histone octamer, around which DNA is wrapped. A typical nucleosome contains 200 base pairs. DNA is acidic and histone is basic in nature. Because of the presence of basic amino acids, lysine and arginine in histones. 31st. It is a polynucleotide chain where A is 5' end and B is 3' end of the chain. 32. This figure is of a replication fork, which is the small opening of the DNA helix. Replication of DNA is continuous at point A, where the polarity of the template strand is 3' to 5', and it is discontinuous in the form of Okazaki fragments at point B, where the polarity of the strand is 5' to 3' end. 33. This is a structure of a sperm, where A is acrosome, a cap-like structure containing enzymes that help in fertilization of ovum. B is the neck region, and C is mitochondria present in middle piece D, which provides energy required for the movement of the sperm. 34. It is a diagrammatic sectional view of a seminiferous tubules, where A are spermatozoa, B spermatids, C Sertoli cells, which provides nourishment to the germ cells, and D spermatogonium. 35. It is a diagrammatic section view of ovary, where A is tertiary follicle showing antrum, B graphene follicle or mature follicle, C ovum, which is released when graphene follicle ruptures during ovulation, and D. Corpus luteum, formed from ruptured Graafian follicle, and it secretes progesterone hormone. 36th, the figure is showing human fetus within uterus, where A is placental villi, B. umbilical cord, C. amniotic cavity, and D. embryo. 37. This diagram shows the plant of Commelina with two types of flowers, F1 and F2. These flowers are: F1 is chasmogamous flower, which has exposed anther and stigma, and F2 is clistogamous flower, which does not open at all. 38. This is the Gynosium of Michelia, which is multicarpellary and apocarpous, means carpels are free here. 39. It is an apple which is a false fruit, a fruit where other parts of the flower instead of ovary takes part in the formation of fruit. Here A is thalamus, which forms fruit and is the edible part of apple, and B is the seed. 40. This is strawberry, which develops from an aggregate of achenes, and the edible part is thalamus. It is thus a false fruit. Here in the diagram, A is thalamus and B achene. So that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.